Thank you, and thank you all for joining us for our presentation, Implementing a Skills-Based Biotechnology Program. As mentioned, my name is Ingrid Miller, and I'm a Product Manager for the Biotechnology Explorer Program. I'd like to take just a moment to ask that if you have any technical questions that you would send a text chat to the host. If you have any questions during the presentation about the presentation, please submit them to all panelists via the Q&A function, and we will try to answer as many questions as possible at the end of the presentation. We want to let you know that we are recording this webinar, and we will send it along with the PowerPoint presentation to each of you following the event. I'd like to introduce you now to Kirk Brown. Kirk is a national board-certified teacher with extensive experience teaching at the high school and college levels. Kirk has been teaching at Tracy High School in Tracy, California for over 24 years, where he currently holds the position of Science Department Chair. He is a key figure in helping students develop into savvy biologists by integrating his teaching with inspiring hands-on laboratory experiences in his International Baccalaureate Biology and Biotechnology courses. Kirk's passion for education extends into and beyond the post-secondary level as an adjunct associate professor at San Joaquin Delta College, where he has taught courses in core biology and fundamentals of biotechnology. His many collaborations between industry and education have led to significant teacher and student advancement. Over his career, he has helped to form the Agricultural Scientific Academy at Tracy High School and has worked extensively at Lawrence Livermore National Laboratories as a master teacher, helping to develop the Edward Teller Education Center's curriculum. Kirk has presented over 350 workshops in 4 countries. He is highly involved in educational technology, where his department integrates various strategies into their everyday teaching. We are happy to have Kirk with us today, who has just authored the textbook, Biotechnology, a Laboratory Skills Course. Welcome, Kirk.
Thanks, Ingrid. So I'd like to tell you a little bit about the agenda for today. So first of all, I'm going to talk to you a little bit about me and where I came from and how I got into teaching, a little bit about implementation strategies and how I got started and things that might work for you, some unique strategies that I've found over time that I've employed in my own classroom, and then how the steps that you might follow to build your own program. So my background begins when I went to college. So my bachelor's degree was in biology with a concentration in entomology, and I intended to actually pursue a career as an integrated pest manager, and so I was accepted to UC Davis to go to graduate school there. In starting that pathway, I actually got a job in the summer, right after I graduated, as a pest control advisor. And during that experience, I was poisoned a couple of times, and it just really wasn't what I thought it was going to be. So I immediately, switched gears, and I decided, I'm just going to teach for a couple of years before I can decide what to pursue in graduate school. So I enrolled in the credential program at University of Pacific in Stockton, California. And initially, they had decided to place me at a suburban school in the same town where I'd grown up, and it's where my dad was an actual teacher. So I thought this was going to be great. Well, the university thought that was not going to be good for me, and so they decided to place me in inner city Stockton. And I don't know if you know anything about Stockton, California, but Stockton was the most violent city in the United States at one point, and they still hold that title. So I was placed in the care or under the tutelage of a master teacher that was a little Puerto Rican woman that was initially, I thought, extremely gruff and had a real rough demeanor. And after being there just a few short hours, I realized that her approach to the students and how she earned their respect and held them to such high standards and really motivated the kids to do something. Now, that says a lot from inner city Stockton, because the students that I had in class that year, many of them were parents themselves. I had one student that had 2 children before she was a freshman in high school, and we had an on-site daycare center there where the students would actually come to the science department, and we had a little lobby area that had live animals that they could look at, and they would go on field trips there. And my students would literally wave at their own children as they came by. So over that experience really showed me a couple of things that I still use today, and that is that you can have very high standards. You can hold kids to that, to have rigor in your curriculum, and the relationship you build with students really helps motivate them. And so I would still be there today at that school, but what happened was at that time there wasn't very many jobs available. So I interviewed and was hired at Tracy High School in Tracy, California. Now, that was very fortuitous. That school actually began in 1917, and when I went there 24 years ago, there was a really wonderful veteran staff there, and they really supported me and encouraged me to be creative and to think outside the box and do new things. So that really started me on a path that led me to where I am today. So I began in '87 at Tracy High, and that was the same year that they actually founded the International Baccalaureate program at Tracy High School. So a couple of years later, they asked me if I would develop the IB biology program at Tracy High School. And that led me down the path trying to look for new laboratories and new experiences for my own students. And I felt like they needed to have some current experiences and be exposed to modern things. So then in the early 90s, there was a movement to try to help the kids caught in the middle. And so I helped found the Agricultural Scientific Academy at Tracy High School, where we used integrated curriculum and project-based learning and a lot of inquiry to help integrate the curriculum and give kids a common foundation. In 1993, I went with a colleague to a meeting at Lawrence Livermore National Laboratory, which led to me applying for and receiving an internship to work in research there. And that was during the heyday of the Human Genome Project. So I really got a wonderful experience there working in research laboratories and learning all about molecular biology. And that's kind of where I learned most of that. And at that same time, the lab was trying to develop a training program for schools and tried to bring that technology out into the public's awareness. And so I helped found the biotech education program there, where we trained entire staffs, entire high schools, many of them across the country even. That was the first foray into large-scale staff development and giving lots of workshops repetitively, and I honed my skills as a presenter at that early stage. During one of those presentations, Ron Mardigian from Bio-Rad Laboratories was present, and he was at the time a product manager at Bio-Rad, and he thought that Bio-Rad had a lot to offer schools and that they could take advantage of the kind of ace hardware of the biotech industry, where the position that Bio-Rad held. And so he had a lot of great ideas. And since that point forward, we've been working closely with Bio-Rad and helping give workshops and training programs over time. In 2001, I started my own biotech course at Tracy High School. It's an ROP class, which is a regional occupational program that's equivalent across the country to career and technical education programs. And at the same time, I became an adjunct assistant professor at San Joaquin Delta College, where I taught biology for majors classes and over time developed a fundamentals of biotech course for Delta College. In 2003, Lawrence Livermore Lab founded the Edward Teller Education Center, and that was a program to help teachers gain enough skills so that when they were placed in a research internship, they could be successful. And so I helped develop a number of programs, one of which was in biotechnology there. And then in 2011, published the Biotechnology Lab Skills course textbook that kind of chronologizes or documents my approach. And so the whole goal here is for other teachers to have the ability to do this in a much shorter timeline. Instead of a 24-year timeline, maybe a couple-year timeline, which would be great. So the big issue is, why write this book? Well, over time, I've taken the internships and the experiences that I've had and actually brought them to my students. And so I've adapted all of those things. So that's one thing that I can bring to the table. Another thing is I've given lots of workshops and talked to a lot of teachers in a lot of different venues, not only with Bio-Rad, but with Lawrence Livermore Lab and the state of California and a lot of different venues. So I have a lot of experience working with people and developing curriculum at all different levels, both site and across the country. I've also had my own course that's unique in the way that I've approached developing my own course, which is I offer at 0 and 7th periods, the hour before school and the hour after school. And I have a level 1, 2, and 3, and it's a mentoring environment where the students that are more experienced at levels 2 and 3 actually mentor and help help the students at level 1 move along. So that's one thing. So one of the things I wanted to highlight is some of my students and how the success that I've had over time has really impacted the students, which I think is an important measure of what's going on here. So Scott Chilton is one of my students, and I had him in International Baccalaureate Advanced Biology 1 and 2, and he was from South Stockton, the same place where I'd done my student teaching years previous. And his dad actually drove him daily to Tracy High School to take advantage of the programs at Tracy High. So he was in my biotech class the hour before school, starting in his sophomore year. So for 3 years, I had him there as well. And as a senior year, he applied and was accepted to MIT, and actually went to MIT. And the summer after his first freshman year, he applied and was accepted at Bio-Rad Labs to do a summer internship, which led him down a path of each summer during his time in high school, or in the university level, to actually explore new opportunities. So he pursued some at MIT itself, in San Diego at the Salk Institute, and then he graduated with his bachelor's degree in biological science from MIT. And then the summer between going to graduate school, he went to Valencia, Spain, which was an opportunity he made connections through his time at Bio-Rad. And then today he's in his third-year PhD program at Harvard University, where he's exploring the proteins involved in bacterial transformation, which is very interesting because some of the very first activities he did was the PGLO lab at Tracy High. So that was really amazing. Well, there's other students that I use as well that are former students of mine, and this is a good example of what you can do as your students move on. Make connections for your kids and make what you're teaching relevant. So here's an example of a student. His name is Nate Leachman, where he graduated from Tracy High, went to Davidson College, left Davidson College, worked at UCSF a couple years in a laboratory, left there, went to UC Davis for his PhD, and now he's a legal advisor to a law firm. And this is one of his publications for the Proceedings of the National Academy of Science. And here's another one of my students, Amr Nijigal. He's actually a pediatric surgeon at UC San Francisco, but he left Tracy High, went to Duke University, where he majored in philosophy and pre-med, left there, went to UCSF in San Francisco, where he started medical school. After his third year, he took 2 years, went to UCLA, did research there for 2 years, back to UCSF to finish his medical degree, where he then went to UCSF for his surgical residency, and now he's doing stem cell research along with his surgical residency as a pediatric surgeon. And then the final student here I wanted to show you is Denise Gangarin. And Denise went from Tracy High to UCLA as a molecular biology major, from there to UC San Diego, from there to Emory for a postdoc, and then from there on to the CDC, where she's associate director of science there at the CDC. So students are very motivated by students that came down the same pathway. And this is a picture of Sophie Wong receiving congratulations from Glenn Seaborg, the only man ever to have an element named after him when he's alive. And she actually won a trip to Stockholm, Sweden to watch the Nobel Prize ceremony. So I guess the teaching tip here is to use your former students in any way you can to give relevance to why they're learning what they're learning, and that make connections. And by the way, Amr Nijigal is coming to my own classroom next Wednesday to speak on National Stem Cell Day, which is a great way to involve your former students. So I guess my implementation strategy is to utilize mentoring environments as much as you can, both currently with your current students, mentoring other current students, but also your past students as well. That really helps. Focus on skill development as a basis upon which, recognized formats and standards can be implemented. So that means, like, I focus on using a lab notebook, and I use that, an industry standard laboratory notebook, where the kids learn how to write and record information in a book, and that really is important. But learning how to use the proper technique is very critical. Have students set up as much as possible. Oftentimes, teachers are so used to putting everything out for student at every single workstation that they think that's the way to do it. Well, sometimes different courses require different approaches, and this is one of those courses. It's a skill-based course. So you want the students to set up as much as possible, make their reagents as much as possible, and to be involved there. So my approach really is to take 3 quarters of skill development and then 1 quarter of project work. And this is what allows them to actually take time to apply what they know and to potentially add inquiry to what they're doing. And oftentimes, you can't really do inquiry until you know the content. But oftentimes, if you try to do inquiry at the same time you're developing content, there's a problem with that. So taking time in a course to do 3/4 of new project, new skill development, and then one quarter of implementation is critical. So do that. The final stage of all research is communicating that research. So teaching students how to make a poster like they would do at a university level, that's kind of like what an introductory researcher would do first, is important. And then there's other opportunities for communication of findings. So in starting your first course, there's a lot of issues, administrative issues you should take into account and think about. One is, will this count for the student's coursework that leads to the university? So in California, we call that the A through G requirements, but each state would call that something maybe slightly different. But those are very critical, because if there's not some sort of connection to that can be an issue. So look into that. You might actually contact your district administrator, whoever's in charge of that program. Even your site principal will probably know how to do that. The courses can be articulated with local community college programs if you're doing this at the high school level. If you're doing it at a community college level, looking to get these articulated with the university. So oftentimes there's even an articulation all the way through. In California, we call those 2+2 articulations from high school to community college. But if you're going from high school all the way, they call that 2+2+2. So there are different types of articulations, and it's worth pursuing those. It also brings other people in the mix to continually evaluate your own program to make sure your current and your skills are also valid and that you're teaching the right thing, which is very important. The impacts to the master schedule. So at the high school level, we certainly understand this, and that is the master schedule is all the courses offered at a school at one given time. And so if you add one to the mix and you add a singleton, one that's only offered one period, that can actually affect how kids are scheduled in their courses. So talking to your principal, thinking outside the box, ways to get around that, and that's what I did, and that's how my course ended up to be 0 period. And also not just how it affects the organization of the school schedule, but what about the student schedule? And are they available? The coursework that's offered at your school, is it making them choose between maybe a theory-based course versus an applied course? And so the way I got around that is to offer mine 0 period and 7th period, and also varied days of the week. So instead of it being every day of the week, it would be either Monday, Wednesday, Friday, Monday through Friday, or Tuesday, Thursday, and they can repeat year 1, year 2, and year 3. So that's a very complicated way of doing that, but it meets the needs of my students and at my school. So I think that's the take-home message there, is just to think outside the box a little bit and think about these other approaches that might help you really make that happen. And then ROP and CTE requirements. So ROP is Regional Occupational Program, but across the country that might be often referred to as career and technical education. These courses fit very well within those programs. You don't have to, but they allow for potential funding sources, and also relate very well to skill development, and that's the approach here and that I am recommending. So the book that I wrote, the Biotechnology Lab Skills courses, is divided up into various chapters, and so I just wanted to give you a flavor of what those chapters might look like. And so Chapter 1 is an overview of the biotech industry. Chapter 2 focuses on the lab skills, just basic lab skills. Chapter 3 is microbiology and cell culturing. Chapter 4 is DNA structure analysis. Chapter 5 is bacterial transformation and purification of plasmids. Chapter 6 is polymerase chain reaction. Chapter 7 is proteins and analysis. And then Chapter 8 is the immunological applications of that. And then Chapter 9 is solely focuses on projects and project ideas and project implementation and communication of findings and things like that. And then there's a myriad of appendices, which I'll get into a little bit later. So the book is really a hybrid book, which it starts out with giving background information on a topic, and it's the background necessary for the students to understand the activities that they're going to learn, the skills they're going to learn. So it's just pared down to be enough that they can understand what they're doing, so not a lot of extra in there. Also, the laboratories are actually in the bound textbook, which is nice because they don't have to have 2 separate textbooks and things to carry around. And the activities or quick guides actually have a pictorial flow down one side. It helps students that are maybe EL or English as a second language, but it also illustrates key concepts of what things might need to look like or what will look like, and that helps students follow the protocol. Oftentimes that's a challenge, and so that's to help with that. And then there's a teacher supplement that goes along with that, and that, as a teacher, these are very important because it talks about what are the anticipated results, what should they look like, how do you analyze results, how would you assess this in your classroom, how do you prepare everything, and how do you couch that, how do you connect it to other things. That's very important. So in the strategies I think that I've developed over time that really work well for me is the focus on laboratory notebooks and the idea that you're learning skills, but you're also learning how to write in a notebook. And then along with that, the most important thing to help analyze the results of that is the use of rubrics. And rubrics are both good for the student and for the teacher because it gives you something to talk about. The emphasis on skills is critical. And then also we have rubrics in there that are specifically describe what the skill is. And the style of notebook I like is novice, developing, and proficient. And that ramifies through these skill-based rubrics. Also, there's rubrics in there for helping students propose a project and what should be included in a project proposal. And there's rubrics for scoring that. And there's also project monitoring rubrics. And what do you look for as the projects are progressing? So one important strategy that I think that you should think about, and that is the use of authentic assessment, which is you're looking at student work. And this is an example of a lab book page where the student would bring up their lab book with the rubric and have analyzed their own stuff, and then you have a conversation with them, and you can point out what they should be there. So this happens to be an entry on making solutions, so I'm looking for units and cross-canceling and things like that. So it's kind of important that this enables that conversation to take place, and students really like that. Each activity should be assessed. Don't skip over any because it gives kids practice talking with you and having you look and point out things that they can improve. So those notebook rubrics are a critical component, and it's been very successful over time for me. So the teaching tip is have students use the rubric the first time to document something simple like washing out how to wash a reagent bottle or how to make a DNA necklace, something that's easy for them to follow that they can just concentrate on writing in the book. Oftentimes we start with the protocol that they're having trouble learning the protocol first, but start simple and then let them build skills over time, and that's very helpful. Another thing that I think is very important, and that's have students use rubrics to self-assess as they do their work, empowers them to keep getting better and keeps improving. And along with that is I let them improve them. So if they come and they're really kind of off the mark, I tell them what to improve, and then they go back and improve that. So this is an example of one of the skill rubrics, and this happens to be how to use a serological pipette with a pipette pump. So you notice that there's three real main categories there: novice, developing, and proficient, and it describes them. And so the teaching tip, the take-home message here, the teaching tip really is have students keep a running list of once they get proficient, to write that proficient skill in the back of their lab book and have a running list. This is fodder for their resumes. And when they get ready to apply to a research laboratory, maybe their freshman year in the university, they can actually use that on their resumes. And that just helps them also kind of check off things that they actually know how to do. So Ingrid?
Thank you. We just wanted to take a moment in between to do a quick process check to remind you that if you have questions about the presentation or for Kirk to answer at the end of the presentation, please submit your questions to the panelists via the Q&A function. If you happen to be having any technical issues right now, please use the chat function to the host. And also to remind you that we will be sending out the replay recording of the presentation and a copy of the slides to each of you following the presentation within a few days. Thank you.
Thanks, Ingrid. So another thing that we've thought a lot about as a teacher, what do you decide is like, what activities do I really do? Well, there's in the teacher's supplement, there's various appendices in there that help you might plan that. So if you are just starting, what activities would be a good list of activities in your starting program? What if you're not just starting and you're down the path a little further, or your students have a little bit more skill set? There's an intermediate list, and then there's an advanced list as well. So that helps get started and helps you think through that without having to start from scratch and just do it by trial and error. There's also pacing guides in there that are for not only high school, and based on if you were in a block schedule or just a straight 6-period day, but also there is a matrix in there if you're a college class. And when I taught college, I went through this as well. So I think there's it gives you a pace that you're not going to be way off the mark to get started. Also, there's relative background information that you should kind of go over that leads into what if you had an introductory course or an advanced course, what are the list of activities that go along with that same pace. So again, this will get you started and get you down the path so you won't have to do all this by trial and error. But the big message here is these placing guides are very flexible. Implement what you can with what you have. So oftentimes teachers think, well, gosh, I don't have very much, so I have to wait until I have everything. When I first started, I had literally one gel box and one pipette, one micropipette. And the students, when I talk to them today about that, they don't think that we only had one gel box. They don't even remember that. They remember doing electrophoresis and the fact that I found a gel box that had a lot of lanes in there. Every student had their own lane, and we shared the pipette. I structured the class so they just stood in line and did it. And so for them, it wasn't that they didn't know any different, right? So remember, anything is better than nothing. Start and get going, and then you can build from there. So in that same light, there is a minimal equipment outline so that if, what if you had very, very minimal things? What could you do? There's a lot of activities in here that don't require a lot of, complicated equipment to get started. Over time, you can work toward that, and I'll talk more about that. But when starting, purchase equipment that you can serve a lot of students initially. If you can, buy enough to get started and then build on it. So try not to buy a lot of different things if you can help it. But when I get into the equipment part, I'll talk more about that. But have a plan of action, so that if anyone asks you what they can do to help buy things for your classroom, you already know what you need, and you thought that out. So that's kind of important to do. In the course, one of I think the unique aspects of my program here is the emphasis on projects, and that this is where the students really solidify what they know. So you go through all of the protocols for about the first 3 quarters, and then at the end, Chapter 9 is about 100 different project ideas. And what I've found over time is with students, what they struggle with is coming up with their own idea. If you leave it up to them and just say, hey, come up with your own idea, there'll be months pondering things because their background level is not quite what it needs to be. So in most cases. So having a list that maybe even is broken down by either content topic, which they're lumped with forensic science or agriculture or molecular biology or whatever it is, but there's different types and levels that are of different complexity. So we use the icons for like skiing. So it's like a green circle, a blue square, and a black diamond. They go from being the green circle would be very simple projects, and here's an example of one of generating bacterial stock cultures. That applies what they learn, pouring plates, doing streak plates, doing things that some of the microbiology skill set that they want to work on. If a little bit more complex than that, the blue square is evaluating mini-prep protocols, a common practice in a research environment, where you're comparing 3 different approaches and seeing what yield you get. So there's a lot of good projects that students don't think are projects. They think you have to cure cancer to be a research project, but really it's not. It's doing good science, recording it, and applying the skills that you've learned in a new way. So that giving the students these ideas gets them started faster, and then that 1 quarter is not going to be wasted them coming up with ideas. So when they get ready to communicate their research, teaching them how to do a research poster is important. And remember, there's different permutations of posters depending on the venue. And so this is an example of one of my student posters on using PCR to determine the mitochondrial sequence of sheep. And they take a flock of sheep and they're trying to look at how the sheep are related to one another. But in different presentations or different venues, you might actually have to do a research paper or even go and give a PowerPoint presentation. And so there are many different venues that students can actually enter their work. But one of the things when students are doing posters and things like that, one of the things you might think about, and that is teach appropriate statistics so that the students are not so simple in their tables. They're not very sophisticated. With just a little bit of extra time, you can teach students about how to use averages and standard deviations and equations of line and things like that, and how to actually label a table. Teach them how to use proper labels for tables and figures, and how to add those captions. That really just it's a little bit extra that makes their posters and papers really pop and really look great. So same thing when using graphs. Most students know how to use basic graphing software and basic graphing skills, but what they don't know how to do is how to put error bars and standard deviations on their graphs, and when to do that, and when to use a linear curve versus an exponential curve or something like that. So taking time to give these skills to your students is just as important as the pipetting skills and the PCR skills and those other skills. They're equally used by scientists, if not more. So take time to do that. So when you're thinking about what equipment you might purchase, this is always, I get these questions a lot. What would you buy if you had, what would you do first? Well, depends on what my objectives were in the course that I taught. So my advice to you is to try to pick something, number one, that's industry standard, and this equipment is certainly industry standard. Bio-Rad being kind of the ace hardware of the biotech industry, it's nice because they will see these same gel boxes and power supplies in the research laboratories when they go to the university level. So my big advice is to do that. Now, if you can't, let's say you only have enough money to buy one gel box, and one mini gel won't quite have enough lanes for your entire class. Well, Bio-Rad has a gel box that you could use, and that's a wide mini gel. And that has 20 lanes across, and you can double stack those and get 40 lanes across in there. So like I did my very first time, I only could afford one thing, so I bought something like that. And you get a simple power supply, and you can now do electrophoresis with your classroom. You might have to share, it won't be optimal. But when you have more money and you get more funds, then going to the mini gel boxes and getting enough for your whole classroom is wonderful, and you can keep this wide mini gel then for research projects where kids might want to run lots of samples in one gel box. So that's really great. Another thought is that initially when you buy micropipettes, oftentimes teachers buy the 2 to 20. But over time, work toward getting the entire set for your classroom so that the students get used to deciding when do you switch gears, when should you use a 0.5 to 10 versus a 2 to 20, when do you use the 20 to 200 and so forth. So that gives the skills another skill set that they're going to generate. Now, other types of power supplies. The larger power supplies, this happens to be a Power Pack Basic, this has a countdown timer, and as a teacher, that's a critical thing, where you can start the gel box running and walk away, and they just count down and turn off automatically. So you're not running samples off the end because you're busy chatting, and as teachers, we tend to be windy and keep going on and on and on, so we don't want to do that. But I guess the message here is really think about a program, and think as soon as you can about what new things you're going to add year after year. Getting started, and then starting to tell yourself, okay, and by next year I want to add this component. By the following year I want to add this component. And as you add components, like one of the things you should do initially as soon as you can is to think about adding PCR, because it's really such a fundamental, protocol or tool used in science, so you should really think about doing that. But oftentimes, teachers maybe have not had a lot of experience using protein gels, and those are just wonderful, and adding Western blots and things like that. But it really just depends on the program that you're trying to implement and the emphasis that you're focusing on, agriculture or whatever. You might be doing a forensic science program. You might just be embedding these in a pre-existing program, maybe at the end of an AP course or in the middle of a biology class or whatever. There's just a lot of different permutations there that you might use these for. So the ultimate question always comes up is, how am I going to pay for this? And there are so many different ways that you can go about, and I've used all these sources, and over time that's just how I funded my own program. And that is one of the things that people say, I'm not close to a university, or I'm not close to a biotech company, so I can't do this. And it's like, well, almost every hospital has an educational foundation that they fund programs in the local communities, and so pursue those and look into that, because even a collection of doctors in town will oftentimes donate things to local schools, and so they definitely would see the need for that. Service clubs like Kiwanis and Rotary and Seroptimist and all these different service clubs are great helps to schools, but you have to get your program together and start thinking about what you might do to what do you need, and put it together as a simple proposal. Grant Wrangler is a website that is like a clearinghouse for different grants, and it's a wonderful website, so check that out. Bio-Rad has a grant writing webinar that's on the webinar list at the end, and you'll see the link to this. But Stan Hitomi, a friend of mine, has done a wonderful job, and he has been very prolific in grant writing, and the webinar he has produced is wonderful because he gives you very concrete examples and things to do in order to be successful in writing either simple grants or even very complicated grants for a lot more money. The NSTA, NABT, any professional organizations have grants available and kind of a current list of grants that are out there, including your local state organization, California Science Teachers, Georgia Science Teachers, Arizona Science Teachers. They all have those as well. So go visit those websites and look at that. And award competitions. Now, oftentimes teachers shy away from award competitions. They think, oh, I'm tooting my own horn and I don't want to do that. You know, that's so typical for teachers. And I feel the same way. But what I found over time is that those award competitions are a really good way of generating funds. And oftentimes there's a lot of money out there for teachers to use in their classroom. And that could be one of the ways that you definitely can seek funding. And there are some that Bio-Rad sponsor as well. So you can see, you go on those, either NSTA, and that's one of those out there. Local industry and business partnerships. So look at what industry is in your area. Maybe it's agriculture, and there's going to have some of these places, maybe a seed growing company, or maybe it's a fertilizer company or something like that. Remember, they rely on having a scientifically literate workforce, and you're going to be helping build those skill sets for those students. So they want to partner with you in helping to ensure that occurs. So those are really important things to be thinking about, and you've got to market that because that's how they're going to do that. So remember that writing grants is really, and looking for funds is great to talk to students about, because remember, every researcher that's ever done any work at all knows all about grant writing, and that is a big critical role that they have to learn and do that. So involving the students in that is also important. I actually have my students write proposals to me and actually have a budget associated with that, and then how would you raise the funds? So I have the students even go on to various places and request donations of very specific things when they get into their research projects. There's a lot that they can do and get involved with. It really helps them learn. So there are many opportunities for teachers. Now this is where I get on things. Where did you learn all this stuff? How did you do it? Remember, I have a 25-year timeline here. We don't want everyone to have a 25-year timeline. So there are other places you can get access to this right away. You can go to science education conferences and talk to people like me, and I can help give that to you as fast as I possibly can. But and those are great ways to get started. Every local university and college has summer programs. BioLink Network, it's a community college network of biotech programs across the country. They have a wonderful website, biolink.org, that you should go to, that has a clearinghouse of workshops across the country that are sponsored by those, and they're really great workshops, so you should think about going there. Bio-Rad has 3 curriculum and training specialists across the country. Sherry Andrews is on the East Coast, Lee Brown is in the middle of the United States, and Damon Ty is on the West Coast. And these people have great experience, and they'll come out and really help you and connect with those people and connect with them electronically or whatever means that you can to get their expertise. That really helps. So Ingrid?
Great, thank you, Kirk. We wanted to give you a little bit of information about how you can get the book. You can contact Bio-Rad customer service within the U.S. to purchase the book. The phone number is 1-800-4-BioRad. There are a couple different options when you're ordering the book. The first to consider is the student edition, which Kirk spoke about, and the other is the teacher edition. The teacher edition includes one each of the student edition and the teacher supplement. Both Kirk talked about what's included in each of those previously. And we wanted to provide a few more resources for you. Kirk had mentioned the curriculum training specialists that Bio-Rad has. You can reach them at biotechnology_explorer@bio-rad.com, or you can go to our website and look at explorer.bio-rad.com as well. We also have an excellent technical support team that's ready and loves answering questions from educators. So they're a very well-educated group of people and can help you with any of your product or scientific question needs. We have a list of other webinars that have been pre-recorded, so if you find this interesting and helpful, we'd recommend that you go ahead and go online and check out some of the other webinars that we have. I think they're very helpful. And Kirk has kindly offered to provide his personal information for you, so his email is listed there as well as his personal website, so he's happy to answer and entertain any questions that you have for him as well. So now we're ready to answer your questions. So some of you have been sending your questions in via the Q&A throughout the presentation. And if you have more questions, continue to send them in as we go into the question and answer session. And I will start off with the first one. So Kirk, the first question is, what is an estimated equipment startup cost of a program like this for a class of 15? I am a PhD high school teacher and an independent school looking to develop a biotech program.
Well, that's a great question. That really depends on the type of course you want to implement. There are many different budgets that you might have at your disposal. The teacher supplement appendix and pacing and purchasing guides, which they're going to be sent to you, has 3 really well-defined courses. There's an advanced one, a basic one, and those with minimal equipment. So, and the minimal equipment one obviously does not include the PCR activities. But these guides actually help you plan your course and think about it. But I guess a ballpark figure is if you're looking at the cost of the equipment they range really from about $4,000 to about $25,000. But really the sky's the limit, as you well know, as you can get into a lot more than that if you get into real-time PCR and stuff like that. But the annual kit cost, if you're going to use these kits, are anywhere between $1,000 and $2,000 as well. That's kind of the basic ballpark. And so really, if you're going to go 15 or more, that's about what you're looking at. So anywhere between $4,000 and $25,000 in equipment and about $1,000 to $2,000 in kits.
Great, thank you, Kirk. The next question is, I just started a biotechnology club that is a continuation of my biotechnology course. The course is content-based, and they do also get the lab skills, but the club is for developing skills needed to get jobs and lab experience. Would you recommend that I try to add a second year of a lab skills specific course too?
Yes, and so that's a great question. And initially, when I first started, I did that kind of thing, is that I had a level 1 course only, and then students came and said they wanted more. And so that's how I started doing level 2. And then level 2 initially was kind of just on their own doing independent things like you're talking about, kind of like a club after school. In reality, what that evolved into over time was a level 2 of the same course, where they go back and deepen their current skills and then actually can, in parallel, be continually working on their independent projects. Because in a real research environment, if you ever go to a laboratory, you see that they have to mentor, but they don't stop doing their own work. And so they have to learn how to balance out mentoring someone else along with keeping another project going. And so it's a great model, exactly the way a research laboratory happens. So not only would I encourage you to make that club a second year, but even a third year, and to model that, because you're definitely you're stumbling on that where I stumbled on a few years ago. And that really is powerful, because if you get kids coming after school for a club, they're already interested, and so you're already made that relationship connection with them, and that they're learning very powerful things from you. So that's what I would do. That's great question.
Okay. The next question is, how can I get the lab book? She'd love to use it with her club, and she also wants to know how expensive it is.
Well, that's a great question. So if you're in an educational institution in the United States, the price of the book is $94. If purchased through a college bookstore, the price is set by a bookstore. The teacher's edition, which is really the student edition plus the teacher supplement, is $120. And if anyone's listening from another country, those countries set their own prices, and you have to contact their local Bio-Rad for that. And what was the second component of that question? There was one about the price of the textbook and
how do they get it.
And how do they get it? So you would order that from Bio-Rad Laboratories directly. So you go to the explorer.biorad.com. Or you could actually email the link to the curriculum and training specialist, and they would help you as well.
The next question is, Bio-Rad doesn't typically do training in Colorado. How can she get us out there? She has a grant and has protein electrophoresis equipment and needs help with it.
Well, that's a good question. If I were you, I would contact Lee Brown at that Biotechnology at explorer.biorad.com. That's probably that's the curriculum and training specialist in that area. I don't know the exact training schedule in Colorado this year. I don't know that we're going to Colorado this year for any of our conferences, but we routinely do go to Denver for various things. So keep your eyes on the upcoming conferences in your area. When they come there, we usually come to Colorado. So, but in the meantime, I would definitely contact Lee Brown. She's definitely in that area. That would help you.
The next question is, does your book include skills with autoclaving and the use of a laminar flow hood?
It definitely includes the autoclave. The laminar flow hood, we touch on those as potential things, but not really. That's not one of the emphasis in this course, and that's one of the main reasons because of cost and of the laminar flow hoods. But in my own course, my own classroom, there are plans online to make your own laminar flow hood, and one of my students made one that works very good. We use it as a sterile hood for doing tissue culture, and so that's one of those extension activities you could build onto your course if you wish to. But know that there's not a whole unit on just tissue culturing for sure.
Thank you. And I wanted to just make a correction. So it is actually the curriculum training specialist in Colorado is actually Damon Ty. But if you send the email to the biotechnology_explorer@bio-rad.com, it'll go to the appropriate person. And the other thing just to mention is we do have professional development training, so that is a paid-for training, and with certain levels of equipment purchases, sometimes that can be included at certain levels. So there's different things that they can work out. So we'll go back to the next question, and the next question is, how do students do an independent project within the classroom, or do you pair them with researchers?
That's a good question. Those independent projects are a combination of both of those. They're done in the classroom. Their final quarter, every student is working on a project. Sometimes they're paired up. Sometimes the students naturally pair up. I don't pair them up. Some students want to do them independently. And then when possible, they'll actually contact they'll start looking into the relevant background information, and oftentimes they'll email researchers, and the researchers will be playing a mentoring role along with me. But everything is done at Tracy High School, at my school, with me. But there are times when they do make those connections, and I highly recommend that. Especially, I mentioned in passing that when they need something, as a particular, maybe a research-based kit, that is something that they use out in the field, like to test for the presence of a protein in a plant tissue or something, then they actually contact that company that generates that and actually ask for a donation, or how would they get it as a student. I make the students do that, and that's really important skill set for them to learn. So yes, partnering with researchers and companies is a very important part of doing research and collaboration. So I definitely recommend that.
The next question is, if the 0 hour and the 7th hour periods are not available, how much should you prepare beforehand at a lab station versus how much should a student do in a 50-minute class period?
Yeah, that's a good question. Oftentimes teachers, we're so used to going fast, right, going as fast as you possibly can, that we think that, okay, we've got to get X, Y, and Z done at a certain point. Well, remember, this is a skill-based course. Very different than maybe you're used to teaching, and I know it was different than I was teaching. So as I first began this, I would do the same thing, go fast. What I've learned is that's not the best way. So what I recommend is that have the students continually do more of that, especially initially, where you need them to pour a gel. Well, they should pour the gel. You shouldn't be pouring that for them. So maybe have them one day come in and do the preparation, and the next day they actually use the materials that they've prepared and actually use that in their own experience. So giving them more of that preparation role, including in my own class, I don't even get anything out. It's put away in the cupboard where it goes, and they actually get it out and put it away, and that's part of working in a laboratory, making things are orderly and put away and stored and carried properly. All of that is part of the skill development that they achieve when they work in a laboratory. So to summarize that, I definitely would say that give them as much that you can. If you're really under a time crunch, you can prep some of that, but remember, every time that you prep that, you're removing that potential practice away from the student. So give them as much practice as you can.
Great. The next question is, if you could choose 3 foundational biotech experiences for a high school student to prepare them for a science major in college, which ones would you choose?
That's a great question. I think I would definitely, I would choose 3. I would choose bacterial transformation, I would choose an experience with DNA electrophoresis, and I would do something in PCR. So, and when I did the bacterial transformation laboratory, I would definitely have the students pour their own plates, make their own augers, you know, dilute everything down, aliquot all the reagents, do the whole thing from start to finish. And then they really understand what it's all about versus setting it all out, all set up, ready to go. That's different. That's maybe the experience they would get in a biology class. But in a biotech class, you want them to really do the whole entire thing. So that does a number of things. It teaches them some aseptic technique when they streak their plates. It teaches them how to, you know, to pipette and just a whole bunch of things. When they do DNA electrophoresis, you can even connect those activities where you take the plasmids and purify the plasmids through a mini prep and actually run those as a lane on your gel as well. But, you know, the electrophoresis experiment and the skills they develop, pouring their own gels, using restriction enzymes, storing them properly, handling them properly, that's just another really wonderful experience. And then PCR is such an industry-based standard that giving them experience using PCR, knowing about that is an experimental technique and that there is things, if you don't follow protocols, it doesn't work very well. And so students need to know not everything is, you know, the procedures and pipetting accurately and stuff like that does affect the results. And so you want to make sure that they learn that. And so that's a good example. Those are 3 main ones that I would choose.
Great. Thank you, Kirk. And then we have another participant who is asking about your reference to the 3 classes that were taught, the Biotech 1, 2, and 3. I'm wondering, with relation to that, what is the level of the book?
The level of the book is really written for either high school students, community college students, or introductory university students. And the book has enough activities in there to definitely easily stretch out over three years by the students revisiting protocols and adding new ones each year. And so that's definitely its purpose, and that is to have a lot of options for teachers. So as they implement the course, they can begin and then evolve over time and actually add new activities from the book. So there's enough activities in there to definitely go for 3 years if you wanted to. So. Great.
And then just time for just a couple more questions. One being, do you need to use Bio-Rad kits to use the book?
The book has over about 18 of the 35 activities are not kit-based. But the reason that the book is aligned to Bio-Rad kits is it does a couple of things. It reduces the need to prepare activities from scratch. And these activities are ones that are complicated to prepare. And speaking from experience, my own self, I certainly use those kits because I found over time doing it all by myself from scratch, there's such variability that the students, when you go through all the steps in doing that, you don't get good results in the end. So it's a combination of things. It's the preparation, it's the quality control that goes into those kits that makes them so robust. And then storing things and stuff like that. They've been QC'd and QA'd so that they really do take a beating and keep working really, really well. So that's the reason why the kits are in there. And they really do work good. But so yeah, I mean, I think you can try to supplement those, but you're back to square one, you know, hoping that the reagents and stuff work well. So so anyway, that's my recommendation, and that's exactly why those kits are in there.
Great. And I think the last question that we have is, do you do plant tissue culture?
I do a little plant tissue culture, and so I teach the basic idea of those plant tissue culture, but then students usually pursue those as projects. So last year I had a student that induced callus and was actually trying to isolate agrobacterium from nature and actually do some work there. And you know, never really got too far on that project, but yeah, I do a little bit of plant tissue culture, just so they understand the basic skill set there.
Well, I think we're just about out of time, and I think we've answered most everyone's questions. So thank you very much, Kirk, and thank you all for joining us for this webinar. And please take a little bit of time at the end to answer the exit survey questions, because we love to know your opinions on things and what future webinars you'd like for us to do. Thank you.
Thank you very much.